Yes.
Yes, Saad, can you hear me?
Yes, I can hear you.
Okay, so the indicators. Give me a minute. Okay, let's start indicators here. Indicators, I told you that we are not using litmus paper in titration, because we have to add drops of indicator. I mean, we have to use some liquid indicators which can tell you the end point of the solution. I mean, they should have different color in acidic solution, they should have different color in a basic solution, and they can tell you that your solution is going to be changed from basic to acidic or from acid to basic. Okay, so I just use few names a few minutes back that we have few indicators in the syllabus. Let's start with a strong base and a strong acid titration. Okay, so if you are using a strong acid and a strong base together, we just discussed that whenever you use a strong acid and a strong base together, you will get complete neutralization. Means there will be complete neutralization with a lot of water molecules. There will be complete neutralization. Okay, but how would you know that all the acid has been reacted with the base? How would we know? For example, I mean, how do we perform the titration? We always take a beaker or the conical flask. Keep in your mind, we never use beaker for the acid. We always use conical flask. Conical flask is that flask which is having very narrow neck, you can say. We always add acid into the conical flask. Let me tell you the reason. Keep in your mind, we cannot use beaker. Beaker is having very wide opening, you can say. We can never use beaker. We always use conical flask for the titration. This is very important point. Why we use conical flask? Because how do we perform titration? We always add acid into the container, then we add base drop by drop. Then as indicator will change the color, you will come to know that there is no acidic particles now. So you have to stop adding base, because you have your all solution have been neutralized. So we always add acid into the container, then we add base drop by drop. Whenever you add base drop by drop into the conical flask, so definitely you have to shake the conical flask or any container in which you are having acid. For example, I have taken 20 milliliter of acid, and now I am adding base drop by drop. So when I add few drops of the base into the conical flask, I have to shake the conical flask so that all the molecules of base could be completely mixed with the acid. So if you shake the beaker, acid can be splashed out, and we are using a stronger acid here. You can see it can damage the whole skin. Acid is a drying agent. It can it can absorb all the moisture of your skin. So that's why we use conical flask. You can see conical flask is that flask which is having very, very narrow neck. I have met, I have met very wide neck here. There is very narrow neck of the conical flask. So when you shake the acid or the base in the conical flask to mix them up, so acid cannot be come out from the conical flask. That's why we use conical flask here. Okay, so yes. So stronger acid and a stronger base is giving you a complete neutral solution. Which indicator is suitable here? Let me tell you. The indicator which is more suitable with the titration of a stronger acid and a stronger base is methyl orange. That is called methyl orange. Methyl orange is that indicator which is very suitable for a strong acid and a strong base neutralization. Okay, why we are using it? Methyl orange has a different color in acidic solution and it has different color in a basic solution. Let me tell you. I mean, we are using a strong acid and a strong base together, no? So definitely We will take conical flask and we will add some acid into the conical flask, and then we will add base. So when you add acid into the conical flask, so you have to add methyl orange indicator into the conical flask. Let me tell you the color of methyl orange in acidic solution. In acidic solution, in acidic solution, methyl orange has reddish color. Means it will make your solution reddish. Acid and the bases are, acid and the bases are colorless. Acid and the bases are colorless. They are transparent solution, colorless solution. For example, I have taken acid into the conical flask, and that is the stronger acid. Okay, so I can add methyl orange into the acidic solution. Methyl orange has red color in acidic solution. Okay, let's start adding base drop by drop. When you add few molecules of base into the acidic solution, that few molecule of base will give you OH negative ions, which will react with the H positive ion, which is produced by the acid. You will add more base because you are using a stronger acid. It is giving you a lot of H positive ion in the solution. Inside the beaker, there are a lot of H positive ion. So you are adding a you are adding drops of base. You are adding base drop by drop, and every drop of base is giving you a lot of OH negative ion to react with the H positive ion. Okay. When all the H positive ions have been reacted by the OH negative ion of the base, what will happen? Now, after this stage, when all the H positive ions from the acid have been reacted with the OH negative ions of the base, what happened at that time? If you add more base now, so that base will give you OH negative ion. But there is, after complete neutralization, when you add more base into the conical flask, so base will give you OH negative ion, and there is no any H positive ion by the acid because all the acid have been neutralized. Now there is no any H positive ion to react with the OH negative ion of the base. So your conical flask will be having basic solution. Your conical flask will be having basic solution now. But everything is transparent. How would we know? That's why we are using methyl orange here. In acidic medium, methyl orange will remain red. So you will, you can see, okay, the methyl orange is giving us red color. It means the solution is still acidic. The solution is still having a lot of H positive ion. Just add more base now. You can continue adding base into the solution. After some time, when all the acidic particles have been neutralized, now the solution is neutral. If you add single drop of the base into the conical flask, methyl orange will change its color from red to yellow. It will change its color from red to yellow. Why? Because,
Wait, can you repeat it?
wait, let me repeat it. Basically, methyl orange is that indicator which can show you red color in acidic and it gives yellow color in basic. Methyl orange is indicator. When you add methyl orange in acidic solution, it will be yellow. When you add methyl orange in when you add methyl orange in acidic solution, it will give you red color. When you add methyl orange in basic solution, it will be yellow. It will be yellow. When you add methyl orange in basic solution, it will be yellow. So right now in the beaker, we have acidic solution. We have acidic solution in the beaker. So when you add methyl orange into the beaker, few drops of methyl orange in the beaker when you are starting the reaction, so methyl orange will give you red color. When you are starting the reaction, the methyl orange will give you red color. Why? Because you have added acidic solution into the beaker. So methyl orange will make solution red. It means the solution is acidic. Now you can add base into the acidic solution in conical flask. Acid is giving you a lot of H positive ions. So when you add base drop by drop into the conical flask, so base is giving OH negative ion, and every OH negative ion is reacting with the H positive ion produced by the acid. After some time, when all the H positive ions produced by the acid have been neutralized by OH negative ions given by the base, you can say. So the solution will be neutral. After this stage, if you add a single drop of base, if you add a single drop of base into the solution, so methyl orange will change its color to yellow. Because now now the solution is neutral. Now there is no acidic particle left for the reaction with the base. So if you add a single drop of base into the solution, methyl orange will suddenly change its color to yellow. It will tell you that your solution was neutral. Now it is becoming basic because now there is no acidic particle, because before this stage there was acid in the beaker and you were adding base drop by drop. So solution is still acidic. When all the acidic particles have been neutralized with the base, so what happened? Now there is no acidic particle to react with the base. Now if you add a single drop of base into the conical flask, methyl orange will change its color from red to yellow. And it will tell you now you are making solution basic because there is no acidic particle left into the solution. Now you are making your solution basic, means the neutralization has been done. This is how methyl orange works. Clear?
Yeah.
Okay. So whenever we have a stronger acid and a stronger base, so in this type of neutralization, we can use methyl orange. That is very, very suitable indicator here. Let's do other type of titration. The other one, we can select weak acid and you can select a stronger base. In weak acid and a stronger base, definitely the base is giving you a lot of OH negative ion. Yes. So we have weak acid and a stronger base. So now the base is stronger, means there will be a lot of OH negative ion produced by the base. And you are using very weak acid, it is giving you very less number of H positive ions. So less number of H positive ion will react few OH negative ion, but there are a lot of OH negative ion. But you are using weak acid, that is not giving you more H positive ion to react with the basic ion. So there will be a lot of OH negative ions left. It means a lot of basic ions left into the solution. So your final solution will be basic. The final solution will be basic in nature, which indicate this is not complete neutralization. This is not complete neutralization, but this is the titration. Sometimes we have to react weak acid and weak base together. You know why we are doing titration or why we are doing neutralization reaction? Not for the production of water. We don't need water molecule. We already have water. We already have a lot of water. We already have a lot of sea water, you can say. We can filter that water for drinking purpose or for anything. Why we are doing titration? Because as a result of titration, we get salt and water. So we are doing different type of titration to make different type of salt. For example, I need a salt that can be made by weak acid and a stronger base only. So I have to do this titration. So but it depends that which type of salt you need here. Okay, so the weak acid and the stronger base will give you basic solution because you are using a stronger base here. So you will get basic solution. Which indicator is suitable here? The acid is weak now. So you will get basic solution. We always use here phenolphthalein. Phenolphthalein. The spelling is PHENOL, phenolphthalein. Let me write it here. Phenolphthalein. This is the commonest indicator, which is very, you can say, less costly in your lab. And it is all time available. You know, whenever you are doing titration in your lab, so you can see they will give you phenolphthalein all the time. Reason? Reason is not that phenolphthalein is less costly and that is all time available. No. Whenever you are doing titration in the lab, you know, they can never give you a stronger acid for the titration. a stronger acid cannot be given to the students now. So they will never give you stronger acid. They will give you weak acid all the time. They can give you stronger base. Bases are not that much harmful. They are not that much risky. But acid, that is not easy to handle. You know, acid molecule can be evaporate sometime from the container, and they can be mixed up into your atmosphere. They can make your environment acidic. You can inhale that acidic oxygen, acidic air into your lungs. That can make your body acidic. So the stronger acid is not easy to handle. So that's why they always give you weak acid. You can you can check whenever you perform titration by your hand, they will give you only one uh, indicator, and that indicator is phenolphthalein. Why? Because they are giving you weak acid. So what happened with the phenolphthalein? Okay, phenolphthalein is colorless in acidic solution. Phenolphthalein is colorless in acidic solution. It is also colorless in neutral solution. Well, you don't have to focus on neutral solution. Phenolphthalein is colorless in acidic solution, but it is pink in basic solution. As your solution turns basic, it will give you pink color. So when you are doing titration with the weak acid, so inside the container, there will be weak acid. So you can add phenolphthalein into the container. It will remain colorless. You will add little amount of a stronger base. So there will be few H plus ion present in the container, so they can react with the stronger base.
Wait, so in an acidic solution, it's colorless?
Phenolphthalein is colorless in acidic solution. It will not show any color. Unless your solution turns basic, it will give you pink color. It will give you light pink color when the solution is less basic. If the solution is more basic, it will show you more darker pink, which could be reddish color. So it is, the phenolphthalein is colorless in the acidic solution. So right now in the container you have weak acid, you can add phenolphthalein into the container. Phenolphthalein will remain colorless. Okay. Now you can add base drop by drop. The base is stronger. You can add a stronger base drop by drop. I mean, weak acid is giving you few H plus ion right now. No? It is giving you few H plus ion. It has given you few H plus ion in the container. Now you are adding base. So for example, I just dropped few drops of the base. So few drops of the base, definitely having few molecules of the base, and those few drops of the base will give me few OH negative ions, and few OH negative ions can react with H positive ions of the weak acid. Okay, now you can add more base. You will get more OH negative ion. Those ions will react with the weak acid again. But the acid is weak. There is a possibility that if you are adding base again now, so the base will give you OH negative ions now again, but there is no H positive ion in the solution, which is available to react with the basic thing. So your solution will turn basic. The whole solution will turn basic. Whenever we react weak acid and a strong base together, we always get the basic solution. So the phenolphthalein will change its color to the pink. And you will come to know, okay, so the solution is going toward the basic side, means we don't need more base to add in the acidic solution, because that is a weak acid. The acid is not fully ionized. It is not fully dissociate into its ion. It is not giving us more H positive ion, so we don't have to add more base now, because the solution is going toward the basic side, because the phenolphthalein is showing you pink color. So when we react weak acid and a stronger base together, we always use phenolphthalein. Phenolphthalein is that indicator which will remain colorless in acidic solution, but it will give you pink shade in basic solution. Clear?
Yes.
Next titration, What is the next titration? We just did, okay, now we can change the acid. We can use a stronger acid and weak base now. And we just discussed that when we use a stronger acid and the weak base now. So we will get acidic solution. Okay, so let's bring some stronger acid and let's bring some weak base now. So weak base will give you few OH negative ions. A stronger acid will give you more H positive ion. So a lot of H positive ions will be there after the reaction, means more acidic particle left after the reaction because you are using weak base. That is not fully dissociate into its ion. It is not giving you more OH negative ion to react with the acidic particle. So the final solution will be acidic. We just discussed few minutes before. Okay. Which indicator is suitable here? The final solution is acidic. Which indicator is suitable here? We can use the same indicator here, which we use with the stronger acid and a stronger base. We use methyl orange. We can use the same indicator here, the methyl orange. Methyl orange is that indicator, which is having red color in acidic medium and that has yellow color in basic medium. You can use methyl orange here. That is the only option. Basically, in our lab, we have only two indicators, the phenolphthalein and the methyl orange. We do have more indicator like bromophenol blue, bromothymol blue, methyl red, but those indicators are not available in our lab. They're not commonly available. So we have only two options, either phenolphthalein or the methyl orange. So when you have a weak acid and you get the basic solution at the end, we use phenolphthalein. Otherwise, we always use methyl orange. So in this case, we are going to use methyl orange again, like we did with the stronger acid and the stronger base. You are using a stronger acid here. You're using a stronger acid here. Achha, let me tell you one thing here. Okay. We are using a stronger acid here. Na? In every case, we had taken acid in the beaker, or sorry, the conical flask. I am using word beaker. Basically, we have to use conical flask. In every case, we have taken acid into the container. But in this case, when you have weaker base, you have to take base into the container. You have to take that weak base into the container. It will produce some OH negative ions, few OH negative ions. Okay, now you can add a stronger acid drop by drop. You can add a stronger acid drop by drop into the container. So as you add a stronger acid into the container, it will produce few H positive ion, and those few H positive ions will react with the basic thing. You can add more drops of acid. This titration is more dangerous, you can say. Number one, you are using a strong acid. Number two, you are not taking acid into the conical flask. You have to acid drop by drop. You have to handle acid more carefully now. You can add more acid, but the acid is more stronger. It will give you more H positive ion. So after few minutes, you can say, after few minutes, your solution will be acidic because you are using a stronger acid. So your solution will be acidic. So definitely we are using an indicator here, and that is methyl orange. Methyl orange has yellow color in the basic solution. We have taken weak base into the container. So methyl orange is showing its yellow color. It is making your solution yellow right now. But as all the hydroxide ions from the base have been neutralized by the stronger acid, and if you are still adding few more particles of a stronger acid into the beaker, your solution will be acidic. But how would you know? Your methyl orange will change its color yellow to red. It will change its color in a second from yellow to red. which is indication that your solution is going to be acidic now. You have to stop adding acid. The given base particles have been neutralized completely. The base is not giving you more OH negative ion for neutralization purpose. You have to stop adding acid now. This is how indicator work. If you're using litmus paper here, you have to use litmus paper after every second here. You have to waste a lot of litmus papers. These indicators are best to use. You have to use few drops of indicator. They will change color in a second as you are adding extra drop of anything into the solution. As the solution is moving from acidic to basic side or basic to acidic side, the slight difference will give you the color change. I mean, here we are using methyl orange, methyl orange. So the methyl orange is turning from yellow to red. So it will give you orangish red color when the solution is becoming less acidic. But if you're adding more acid and more, so the solution will become darker red. It means the whole solution is acidic now. You have to do neutralization, but you have done your solution acidic. So this is how we do the titration. These are the indicators which we have to use. You have to focus on only two indicators. But what happened when we use weak acid and the weak base? Which type of indicator we have to use here? Weak base and weak acid titration. We just discussed that weak base or weak acid titration will not give you the proper result, you can say. If someone asks you that which indicator we have to use when we are reacting weak base and weak acid together, you have to give one answer only. No indicator will be used. Why? Because weak base and weak acid, they are not suitable for any titration. Why I am reacting weak base and weak acid together? To make water molecule? No. We already have 75% of water on the earth. Then why I am reacting them? To make a salt? Okay. But they are not reacting properly. They are not giving me ions properly. How will I get salt? I'll get 1 or 2 or 3 molecules of salt, which are useless for me. So the weak acid and the weak base. We can discuss weak acid and the weak base neutralization theoretically, but not practically. And titration is a practical process, an experimental process. You have to do titration by your hand. So the weak base and the weak acid is very good for neutralization reaction, but not good for, not suitable for the titration. So we are not doing titration with the weak base and the weak acid. So we are not using any indicator here. So this is all about the indicator and the weak acid and the weak base. Is there any confusion?
So wait, if there's a weak acid and a strong base, what method is suitable?
When you have, oh wait, what question you just asked me? Weak acid and strong base?
Yes, weak acid and strong base.
Weak acid and a strong base, you have to use phenolphthalein. You have to use phenolphthalein because weak acid and a stronger base, weak acid and a stronger base will give you basic solution. Weak acid and a stronger base will give you basic solution because base is a stronger. So you have to use phenolphthalein. Phenolphthalein is that indicator which can show you pink color in basic solution, and it will remain colorless in acidic solution. I mean, if you are doing titration with weak acid and a stronger base, so the end point will be there when your solution is going to be basic. It means your end, I mean, this is the end point of the titration. You have to stop adding more base here. So you have to use phenolphthalein. You know, phenolphthalein is, you can say that is very strong indicator. It changes color in microsecond. So phenolphthalein will give you pink color as your solution is going to be basic. So when you have weak acid and a stronger base, you have to use phenolphthalein indicator, which will remain colorless in acidic solution, and it will show you pink color in basic solution. And when you are using a strong acid and a strong base, or a strong acid and the weak base, you have to use methyl orange. There are only 2 options in your syllabus for the indicators. You don't have to discuss the thymol blue or the methyl red. Nothing is included in the syllabus. Clear? So this is all about the acid base. So you can go through with the with your notes or something. If you still find difficulty, you can tell me. We will discuss that in the next class. Okay.
So if it's a
Yes.
just one last question, if
Yes,
it's a
yes.
strong acid and a strong base, which method is suitable?
Wait, a strong acid and
Strong base.
a strong acid and a strong base. That was the first one. There will be complete neutralization. A strong acid. Wait, let me write it here. A strong acid and a strong base. And the second condition was one more, when you are using a strong acid and the weak base. In both the condition, we have to use methyl orange. We have to use methyl orange. In both the condition, we have to use methyl orange. Methyl orange will be red in acidic and it will be yellow in a basic solution. In both conditions, we use methyl orange, but we are using phenolphthalein when you're using weak base. When you're using weak acid and a stronger base, then you have to use phenolphthalein. Otherwise, you have to use methyl orange only. Is it clear?
Wait, so if it's a strong base and a strong acid, you use both?
A strong acid and a strong base, you have to use methyl orange only. Methyl
Okay.
orange. A strong acid, weak base, methyl orange. You have only one case in which you have to use phenolphthalein, that is weak acid and strong base. Otherwise, you have to use methyl orange. A strong acid, a strong base, only methyl orange is the option. A strong acid and the weak base, the methyl orange is the option. Is it clear?
Yeah.
So this is all about today. You can go through with your notes. If you still find difficulty in this topic, you can ask me, or you can send me question through WhatsApp, or we can discuss in upcoming class if you have more difficulty. Okay?
Okay.
Take care. And Hafiz.